Hello viewers, welcome to this particular lecture on microbiology. In this particular lecture, myself and my colleague Dr. Sudeshna Shamchaudhuri will be discussing the different experimental basis by which the microorganisms can be isolated from food samples. In this lecture, we have selected three different food samples, namely curd, then pan masala and bread. There may be different microorganisms growing in the air and on these food samples. So the contamination can be endogenous or it can be exogenous. So in this particular lecture, the introduction will be given by myself and the experimental basis will be shared by Dr. Shamchaudhuri. Curd may contain different host of bacterial organisms, of which there may be lactobacillus, there may be lactococcus, there may be pediococcus, there may be streptococcus. But lactobacillus is the most important organism present in curd, which is gram-positive in nature, it is rod-shaped, and at the same time it is catalase negative. So it can easily be identified. This particular bacteria was called to be a probiotic by Mechnikov in 1870. Why probiotic? Because it is helpful for living organism, including human, and as it is present in human gut, it is helping in the colonization of other friendly bacteria. And this particular bacteria also initiates the cardinning reaction of milk. How? Actually, it brings about the coagulation of the lipid substances present in the milk and it produces lactic acid, so it makes the pH very much acidic. And the lactic acid bacteria may have got some different role. It may be important as probiotic, it may be useful for the production of dairy products acting as a starter culture. It may also have importance as an antimicrobial agent, or it may produce exopolysaccharides, it may produce different vitamins, enzymes, and thereby it is very much useful in medicine as well as in food industry. And of course, because the pH goes down to 2 to 3, naturally one particular species, which is Lactobacillus acidophilus, which is most commonly growing in curd. The particular organism may have different advantages and disadvantages. Advantage is that it is used in curd making, it is used in pickling, it produces vitamin B, it can be anti-cancer, but at times it can be a particular organism which can spoil different alcoholic beverage like beer. Now this particular bacteria belong to the family of Lactobacilliaceae along with Bifidobacterium, and broadly there are three different species like the Lactobacillus acidophilus, Lactobacillus plantarum and Lactobacillus rhamnosus. This particular bacteria can be used as biosurfactant. It can produce different organic acid, it can produce protein like bacteriocin, preventing the growth of other bacteria. It can also help in co-aggregation of different other probiotic bacteria, and at the same time it can actually generate the hydrogen peroxide or superoxides, may induce damage. Not always it induces damage. Broadly, this particular group of bacteria in curd can be divided into three groups. One is the obligatory homofermentative, which include the Lactobacillus acidophilus. Then we come to facultative heterofermentative, the group 2, which include the Lactobacillus casei, and obligatory heterofermentative or the group 3, which include the Lactobacillus brevis. And they grow in the milk and they bring about the cardinning reaction, and these are the colony aggregate in the curd which can easily be stained. And you can see that this is not only the Lactobacillus, there may be other species as well which can tolerate low pH.
So sir, which type of species will prevail after the curd formation has been initiated?
Because the pH has gone down to 2 to 3, so mostly the acidophilic organisms will grow, and that mostly include the Lactobacillus acidophilus and also the Streptococcus lactis. So these are the two major organisms which can grow in that low pH. Okay. Now, I already told you there may be other organisms growing which may include the Weissella, Siberia, And normally these are not that much helpful, but they can also bring about the cardining reaction. They are friendly group of bacteria. All these bacteria, that is the Lactobacillus casei, Acidophilus, Salvarius, Thermophilus, they all are protecting the gut, preventing the growth of pathogenic organism. So that's why they are probiotic in nature and which can be experimentally proved. Of course, the probiotic action is mostly compete for nutrients. So they are not allowing the pathogenic organisms to grow. They produce specific organic acid which may be harmful for others, or bacteriocins which may be harmful for others. At the same time, they may adhere to the host epithelium so other pathogenic organisms cannot grow there. And of course, they may actually aggregate with pathogenic bacteria and prevent their replication. So thereby, in one way, the increase in lactic acid bacteria will prevent pathogenic attack in our intestine. So curd is good for us. Of course, as I told you that this particular bacteria, the lactobacillus, on one side it is biosurfactant, it may produce organic acid, it can produce bacteriocins, and it is useful to us. It is not a food material though, but we take it along with other particular betel leaf. And this pan masala can be synthetic or it can be natural. It can be natural in the form of areca nut or betel nut, and this is how we make a mixture soaked in particular natural extract and we take it with the betel leaf. Typically we call it pan. So this is the betel nut, and you can see there are particular colonies of mold growing because of moisture condition of these betel nuts. These molds can grow or bacteria can grow. And of course, you can see the entire country has banned the use of this particular gutka or pan masala. Gutka is the common name or branded name given to this pan masala, which is artificial in nature, but of course prepared from this natural material. Except Karnataka and except Meghalaya, all other states, and of course Jammu and Kashmir, all other states have banned the use of this particular pan masala.
So pan masala we know aids in digestion, then how is it cancerous?
Yeah, actually it aids in digestion, that is a notion. Whether it does or not, yet to be proved. But of course, it can cause cancer, which is proved because it actually activates the epithelial growth promoting factor, as a result of which the rate of cell division in the epithelial lining increases, and thereby oral cancer is very common when you are using pan masala. It causes irritation and the cell is changing in texture and rate of division. Third important food, which is bread, and this bread you can see is contaminated with different microbial organisms, mostly they are fungal spore. Because this bread is enriched in starch or carbohydrate, fungal spore will grow and multiply on the surface of the bread, and there may be bacteria also growing which we normally do not always consider because there is overgrowth of the fungi. And these bacteria may include the Bacillus, Bordetella, the Clostridium, Escherichia, the Spirulina, Staphylococcus, Streptococcus, Salmonella, different bacteria. But because of the excessive growth of the fungal spore, we normally do not consider this bacterial contamination, but which may always coexist.
So students, from the previous part of the lecture, it is already known that the microorganisms can be collected from different food material. It may be endogenous like lactic acid bacteria from curd, or exogenous like pan masala, because it is pre-soaked in some water. Okay, so let's see how all those microorganisms can be isolated as well as characterized from different food material. Curd is made by the fermentation of milk sugar lactose to lactic acid with the help of lactic acid bacteria, which can be isolated in some specific selective media as well. Materials required: the curd itself, specific selective media like Man rugosa sharp agar or MRS agar, gram stain, 3% hydrogen peroxide for catalase assay, Daham's tube, 0.3% arginine phosphate buffer saline PBS at pH 7.2 as well as pH 3, bile salt like sodium deoxycholate or taurocolate, nutrient agar, disc as well as sodium chloride in different concentration. First, the collection of sample. The sample is collected from vendor in the very aseptic condition to avoid degradation as well as cross-contamination. Next, sample preparation. The card is mixed with the saline water and it will be stored at minus 80 degrees centigrade by mixing with glycerol. Now, when it will be needed, the card will be taken for streak plate as well as spread plate method to get the pure colonies. Okay. Next one, isolation of lactic acid bacteria from card. So, from streak plate pure culture, the colonies will be taken and will be streaked over the specific selective MRS agar medium. Okay, either agar medium, solid or in the broth of MRS. Physiological and biochemical characterization that can be done with the help of gram staining, catalase assay as well as sugar fermentation, etc. Because if it has the potency to ferment sugar, so within the inverted Durham's tube, it will produce the bubble by designating the carbon dioxide formation as well as different other tests. Then identification of the isolates with respect to the probiotic properties. We all know lactic acid bacteria have probiotic characteristics. First, the isolation of lactic acid bacteria. So, transfer from MRS agar plate. Okay. So, by transfer, it will be taken for the gram staining to characterize the nature of the colony. Okay. Next, the catalase test. To assay the catalase test, taking a loopful of colony, okay, over a slide and 3% H2O2 will be given. If it produces gas, so it will be manifested as the effervescence. So, it can be indicated whether it is catalase positive or not. Now, probiotic properties of the isolates can be characterized by all those experiments. Resistant to low pH from the MRS agar medium that will be incubated within the MRS broth that will be taken for the centrifugation. So, the cell will be precipitated. That particular cell pellet will be resuspended in the PBS saline pH 3 and that will be kept for 37 degrees centigrade 24 hours. Now, the growth will be monitored by spectrophotometric observation by taking OD at 620 nanometer. Next, the tolerance against bile salt. In the same MRS broth, 3% bile salts will be added and after incubation of some hours, again the growth will be monitored via optical density measurement at 620 nanometer. That is the growth observation. Antibacterial activity. Now, the 0.2 micrometer of the discs will be pre-soaked with the MRS broth containing the specific culture and will be placed over the nutrient agar medium. And the disc diffusion study will tell you about the antimicrobial activity of the particular isolates. And the organism against which the antimicrobial activity will be studied are the E. coli, Staphylococcus aureus, Pseudomonas aeruginosa as well as Bacillus subtilis. Next one, arginine hydrolysis test. 0.3% arginine and 0.2% sodium citrate will be taken within the MRS broth and the ammonia production will be detected by Nessler's reagent. So, all those tests are very specific for the probiotic characters. And growth at different temperatures will be monitored starting from 10 degrees centigrade, 15 degrees centigrade, 30 degrees centigrade, 37 degree as well as 45 degrees centigrade. Again, the growth will be monitored and will be tabulated, okay, and in different sodium chloride concentration starting from 2%, 3%, 4%, 5% as well as 6.5% concentration. Now, isolated lactic acid bacteria from the card will be confirmed by having catalase negative Okay, so you cannot see any effervescence, as well as gram-positive bacilli will be observed under microscope. Next, probiotic properties, resistance to low pH. So you can observe very efficient growth in very low pH like pH 3, because you know that lactic acid bacteria can withstand very low pH when it exists within the stomach. And antimicrobial activity is observed against all the microorganisms like E. coli, Staph aureus, Pseudomonas aeruginosa except Bacillus subtilis. Temperature sensitivity, that I have already told you, that will be tabulated. Okay, if you have different isolates which can be designated as C1, C2, they will show differential growth character, but you cannot observe growth at 10-15 degrees centigrade, and you will observe the optimum or maximum growth at 30 degrees centigrade. And interestingly, it is observed that it can tolerate sodium chloride up to 5%. You cannot observe any growth for any of the isolates at 6.5% as well. And it is very effective to tabulate the antimicrobial activity with respect to the measurement of the inhibition of zone diameter. So, inhibition zone diameter can be compared with respect to different isolated isolates. Okay. So, in this way, the lactic acid bacteria can be isolated from the card, which are the endogenous. From the previous part of the lecture, it is known that the pan masala are contaminated by the water, surface contamination. Okay, so you have to isolate the natural microflora, normal microflora from betel nut, as well as moistened erica nut, which is marketed as pan masala. The materials required, it includes pan masala, nutrient slant, forceps, sterile water, laugh, spirit lamp. Laugh means laminary flow. Very good. First, the pan masala will be pre-soaked in sterile water. Then directly with forceps that will be transferred over the nutrient slant. The slant will be incubated at 37 degrees centigrade at 24 hours. Then the growth will be observed. Now, the identification of the bacteria microscopically. Under the microscope, taking from the nutrient slant, the bacteria will be characterized. And it was found that the rod-shaped bacteria having average length and breadth as well. So, all the length and breadth will be measured. It is also known that bread can be contaminated with the fungal spore, aerial spore, as well as with molds. Okay, and that particular fungal specimen can be isolated, can be identified, as well as characterized according to the spore characteristics, according to the colony characteristics, and on the basis of its reproductive characters. Let's see what are the materials needed to do so. A piece of contaminated bread. PDA slant, that is very specific for fungal culture, growth as well as isolation. Laugh, again laminar airflow, as well as spirit lamp is needed. A piece of contaminated bread along with the specific colony will be transferred over the PDA slant, and that will be incubated at 30 degrees centigrade overnight. Okay, and the colony characteristics again will be followed. The result shows that the colony color and the spore characteristics. You can follow the black round conidia that will be observed, that can designate the presence of, maybe the presence of Aspergillus species, because the bread can be frequently contaminated with the aerial spore, molds, Aspergillus species. So students, from card, lactic acid bacteria can be isolated. From pan masala, as surface contamination, some specific gram-positive organism can be isolated. Okay, as well as from the bread sample, black Aspergillus mold can be isolated. By experiments, you can isolate them off, you can characterize them off, you can identify them.
So, from these three experiments, we have seen that card is showing the growth of lactobacillus, which is endogenous in nature, whereas pan masala is contaminated with some airborne gram-positive bacteria, and finally, bread has been contaminated with the airborne spores of mold, which in this case was identified to be Aspergillus species. Thank you.
Thank you.